Hello, are there any questions? Let's give students a minute to log in and I will get going. Alright, let's begin. Can somebody confirm that you can hear me? Great, thank you. Let me get to the video. Share the screen. Let's move to where we were. Come on. I should probably delete that and save. Alright, so what we're doing today is chapter 13, viruses, viroids, and prions. We'd started that lesson last time. There is no lab today, so I will be there from 6:30 to 6:45 to answer questions. But after that point, I will be logging off. So if you have questions, make sure you log in from 6:30 to 6:45. And your unknown project is due Saturday, June 12th. All right, any questions about the schedule? Not hearing anything. Oh shoot, let me get that to save. We talked about the consequences of lysogeny last time, and that is mainly when a cell, a bacteria cell, has a lysogenic phage in it, it has new DNA in the cell, which can give the cell new properties, changing the phenotype of the cell. So we're going to move on to animal viruses, viruses that infect animal cells, and of course all of the human viruses, or the disease-causing viruses of man, are animal viruses. The multiplication of animal viruses follows basically the pattern of bacteriophage multiplication, but there are several differences, and you don't need to know all of the differences. We will talk that there are differences about release, so maybe you should know that one. But their attachment, their penetration, their encoding, their biosynthesis, and the maturation can be different than a bacteriophage. Their release is different, and that is there are some animal viruses which follow the lytic life cycle. Say they are a lytic virus. I think the polio virus, don't quote me on that because I'm not an expert on polio, but I think it is a lytic virus. And it ruptures the cell, lyses the cell, to release the viruses. And there are animal viruses that are lytic, but there are also other ways that animal viruses can live. There's the budding life cycle, and we'll talk about that, where the virus replicates in the cell, and then it buds out of the cell. And when it does that, it mostly does not kill the cell. The envelope viruses follow the budding life cycle, and we will learn why in just a few slides. There are a few other life cycles, but we're not going to focus on them. They're very similar to the budding life cycle. The important part is this virus replicates in an animal cell, and generally it does not kill the animal cell. Usually, if the cell dies, it is because the immune system recognizes that that cell is infected with a virus, and then the immune system goes in and kills that cell. The virus usually does not kill that cell. Is there a question? I'm hearing a little bit of noise. No, I'm not hearing anything now. Maybe that was something in the background. Multiplication of animal viruses follows the general pattern of viral multiplication for the bacteriophages, but there are some differences. You mainly just need to know that an animal virus can replicate in the nucleus, or they can replicate in the cytoplasm. A bacteriophage, of course, can only replicate in the cytoplasm because that is the only portion of the cell in a bacteriophage or in a bacteria cell. But an animal cell can replicate either in the nucleus or in the cytoplasm, and that's because animals have a nucleus, and all eukaryotes do. We're not going to talk about other viruses that affect other kingdoms. We will briefly in one slide talk about plant viruses, but generally we're not going to talk about other eukaryotic viruses, just animal viruses. And we're talking about animal viruses because they're the ones that cause human disease. My face looks a little funny. Is that a wrinkle on the face? It might be because I slept there and there's a mark there. Or it could be because there's just a... must be vain. I'm concerned about that mark on my face. Anyways, the multiplication of animal virus follows the multiplication of a bacteriophage. There are some differences. The virus is different in that it doesn't attach to the cell wall because animal cells don't have a cell wall. The virus will attach to the cell membrane, and then it's brought into the cell either by penetration or by endocytosis or fusion. Fusion is that the envelope of the virus fuses to the cell membrane, and then they become one. And I think I've got a little picture of that. And then they become one, and then the virus is just dumped into the cytoplasm of the cell. Endocytosis happens where the virus binds to the cell, and then the cell begins invaginating around the virus. And then this invagination will be brought into the cell, and the cell membrane will close around the virus, creating, what do they call that? A phagosome. And then the cell membrane will reform. So right here, the cell membrane pinches off, and it creates a vesicle, but we'll call it a phagosome. And then the cell membrane reforms. And that's endocytosis. Fusion. Oh no, this is an actual electron microscopic image of, I think it's endocytosis in an electron microscopic image. Nope, it's fusion. What happens is that the envelope of the virus fuses, which isn't shown, fuses with the envelope of the membrane. And this looks like a phagosome. That's why I was looking at that. So this is fusion, this is phagosome. And then the envelope is brought into the cell, and the envelope is removed, and then the virus is released. This one, when the envelope of the virus fuses to the cell membrane, they just become one, and it's kind of hard to see it. But what happens is the cell membrane fuses to the viral envelope, and then the cell membrane goes around the virus, and then this is open to the cytoplasm, and then the virus just falls into or moves into the cytoplasm. Let's talk about another way that animal viruses differ from bacteria, phages, and that is animal viruses might multiply using the budding life cycle. Now, animal viruses can rupture the cell, and that's the lytic life cycle. And the animal viruses that follow the lytic life cycle, they are non-enveloped viruses. And if you remember, we call those naked viruses. All they have is a protein coat around the genome of the virus. But the enveloped viruses have a protein coat and then a lipid bilayer around the protein coat. And what happens is the virus multiplies in the cell, and then the virus nucleic acid gets put inside the viral protein coat, and that's what we're looking at here. And then this virus moves up to a membrane. It could be the cell membrane, as shown here. It could be the nuclear membrane, or it could be the membrane of the endoplasmic reticulum. What membrane it is depends on the species of virus, and different viruses will gain their envelope from different membranes. I don't know, but I'm thinking most viruses that are enveloped, I'm talking about animal viruses that are enveloped, probably gain the envelope from the cell membrane. And the virus comes up to the cell membrane in a region of the cell membrane that has viral proteins in it. And these will be like the spikes that are made by the virus, or they could be just, what do they call those? Well, proteins, but I was trying to think of another term. Anyways, they will be viral encoded proteins in the membrane, and the virus will come up to that region and then push on the membrane, creating a bud, and continue to push on the membrane until it pushes out, I don't know, the bud, and the membrane will rip off, and then it'll seal, where the membrane will reanneal and reform the cell membrane, and then the envelope virus will have the membrane form around the virus, and then this membrane will form the envelope around the virus. And it won't entirely be molecules made from the cell, because like I said, in this membrane will be largely proteins or lipoproteins or maybe glycoproteins that are made by the virus. And these will be like important attachment molecules for the virus, or the spikes for the virus, something like that. Anyways, that is how an envelope virus obtains its envelope, and that is it literally rips off a portion of the membrane of the cell that it has infected. And that membrane can be the cell membrane, as shown here and here, or it could be the nuclear membrane or the membrane of the endoplasmic reticulum. Any question about budding viruses? Remember, when a budding virus replicates, it's doing so at usually low numbers, and so the virus is shed from the cell at low numbers, and the cell can survive the loss of this little bit of membrane. That this cell usually will not die from the budding virus. Any question about any of that? All right, if no questions, let's move on. I've mentioned that viruses have different genomes. They can have a DNA genome, and that genome can be double-stranded or single-stranded DNA. The genome can also be double-stranded RNA or single-stranded RNA. And if it's single-stranded RNA, you have to state whether it's a positive single-stranded RNA or a negative single-stranded RNA. Each of this virus needs to have a slightly different mechanism for multiplication and biosynthesis, because a different molecule is being made as the viral genome. Any question about any of that? That line moves a little bit on my forehead. That might be just the way it's being made in Zoom. All right, let's talk about the DNA viruses. These are animal viruses that have DNA as their genome. There are 7 DNA animal viruses that cause disease in humans. And you do not need to memorize all of this table. We will talk about the herpes viridae and the pox viridae. I think most of the others we're not really going to talk about. Oh, we'll talk a little bit about the adenoviruses. Okay? Anyways, it lists the different families of viruses, the different type of DNA molecule, and whether it's an envelope virus or a naked virus. And like I said, you do not need to memorize this table. And then it talks about some of the diseases caused by these viruses. If you don't know, Pox viridae is the family that smallpox falls in, as well as cowpox. Herpes viridae, of course, is the herpes virus, like herpes simplex type 1 and type 2. And this also includes chickenpox and shingles. Epstein-Barr virus, we won't talk about that. Mononucleosis. Roseola virus. That was what I was talking about, and I couldn't remember roseola is a disease that infects over 95% of humans, and generally a baby gets roseola. It's a skin rash and the child gets sick. I think they have a fever for a day or so. But they get it early in life, from 3 months to something like 9 months old. And generally, people get it before they're a year old. And it's believed that the parents are shedding the virus and infecting the baby. Of course, it doesn't cause any disease in the parents, but the baby is getting roseola from their parents. All right, let's move on. So we're looking at the adenoviruses and the herpes viruses. These are DNA-containing viruses, and the DNA is generally copied in a DNA virus in the nucleus. And this will usually use viral enzymes, although everything an animal cell needs for replicating DNA is already in the nucleus. So, as a rule of thumb, DNA animal viruses replicate in the nucleus. Any question about that? Once the DNA enters the host nucleus, one of two things can happen. The DNA can form a provirus, and that is where the viral DNA genome integrates into the host cell's DNA. The recombination of the viral DNA into the host cell DNA is permanent, meaning a provirus will remain in the host cell's DNA for the life of that cell. The provirus is permanent and is never excised from the host cell's DNA. How the viral DNA will replicate in the provirus, life cycle, provirus replication, is the provirus will replicate when the host replicates its DNA. Any question about that? So that is similar to, but different from, lysogenic bacteriophage. And that is the provirus will permanently remain in the host cell's DNA, and it will never come out. And well, it'll never leave the host cell DNA. It can replicate when the host cell DNA is replicated. The viral DNA can also be expressed producing new virions. So the DNA will be expressed, the viral DNA, and the capsid proteins will be made. The viral DNA will be copied and will be inserted into the capsid, and then new viruses will be replicated. And that's probably shown in the next slide. So the double-stranded DNA, we're only talking about that virus now because it's just easiest to talk about, will be replicated, new viral DNA. The viral DNA will be transcribed into viral messenger RNA. It will move into the cytoplasm and be translated into the capsid proteins. The capsid proteins will get together with the replicated viral DNA to make the progeny virions, and then this virus can leave the cell. And that could follow more than one life cycle, but generally it follows the budding life cycle. Let's see if I can shrink this, or maybe I can blow it up. Slideshow. So let's look at the multiplication of the DNA virus in more detail. The DNA virus, and specifically it's papovirus, but we're just talking about DNA virus in general, will attach to the cell membrane and be brought into the cell. The viral DNA will be translated into viral messenger RNA. The viral messenger RNA will leave the nucleus, go into the cytoplasm, and be translated into viral proteins, specifically the capsid proteins of the virus. In the meantime, the viral DNA is replicated. We have more than one copy now. And then the viral DNA will get together with the capsid proteins to make the new virus. And then the newly replicated virions will be released from the cell. This can be released from the lytic life cycle, which is shown here, or it could be released by the budding life cycle, depending on the species of virus. Any question about any of this? All right, if not questions, I'm going to move on. Yeah, I shrink that down a little. Here, that didn't shrink. Why is that not shrinking? Down here, I'll do it down here. Let's look at the animal RNA viruses. Bring this up. There are more of them, you'll notice. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 animal RNA viruses that cause disease in humans. The Picornaviridae you actually are familiar with. This is the #1 family of viruses that causes the human colds. The herpes viruses, we've talked already a little bit about them. Coronaviridae, that's the one that causes COVID-19. We'll talk about that in about 3 slides. The retroviruses, we'll talk about that in 3 or 4 slides. The Paramyxoviridae causes a number of very common childhood illnesses. Now, it is true we have vaccines for all of these common childhood illnesses now, but that includes, actually it includes colds, but respiratory infections. I'm trying to remember what is that called, where the child has a seal-like bark. What's the name of that disease? I can't remember. But measles, mumps, and that other very common respiratory disease that children get, and it gives them a cough that sounds like the bark of a seal. Oh well. Like I said, Paramyxoviridae, very common childhood illnesses. Orthomyxoviridae causes the flu. So if you've never heard of Orthomyxoviridae, if you ever get the flu, that's from the family of Orthomyxoviridae. And I think that's the last one I'll mention. But like I said, there are 15 families of RNA viruses. And I went the wrong way. You do not have to memorize this table. It just is introducing the different family names, what the RNA strand is. Oh, there's a double. I knew there was one that was double RNA. And then single-stranded, and whether it's negative single-stranded or positive single-stranded. Whether it's a naked virus or an envelope virus. And then the diseases they cause. Let me shrink that down a little bit. All right, so we're looking at some of the RNA containing animal virus, and you'll note they are very different in their shape and in their makeup, as well as their size. Like this is pretty small, and this is 10 times larger, so is that one. But you can see these are similar in size, but totally look different. All right, let's talk a little bit more about the multiplication of RNA viruses in more detail. Most are, we're just going to say a rule of thumb for RNA viruses is that they replicate in the cytoplasm. So an animal virus that is an animal RNA virus multiplies in the cytoplasm. And that's because all of the machinery for replicating the virus is present in the cytoplasm. Any question about that? Now, with that said, that rule of thumb is correct, but there is one exception, and that is the retroviruses. The retroviruses are an RNA virus, but they replicate in the nucleus. And the retroviruses are different, and we'll talk about them in about 3 or 4 slides and why they differ. And when you look at their difference, you'll see why they are replicating in the nucleus. And that is, their main difference is they have a DNA intermediate, and that's why the retroviruses are replicating in the nucleus. Anyways, the multiplication of RNA viruses is similar to the replication of the DNA animal viruses, but obviously they have an RNA genome, and the RNA replicates generally, as a rule of thumb, in the cytoplasm, the exception being the retroviruses. We're going to look at 2 examples, the positive single-strand RNA animal virus and the negative single-strand RNA animal virus. Now remember, the positive strand RNA genome can be read by a ribosome. The negative strand RNA viral genome cannot be read by a ribosome. And the negative strand is the antisense to the positive strand, meaning these 2 strands are corollaries to each other. Any question about any of that? So for a DNA double-strand virus, the replication is very simple. You just replicate the DNA, and if it's double-stranded, you just make it single-stranded and then build one strand on the single strand. You build the new strand on the single strand, and you build another new strand on the other single strand of DNA. And then you transcribe the DNA to get viral messenger RNA, which will move into the cytoplasm and be translated into the viral proteins, and then the viral protein will get together with the newly replicated viral DNA. For a positive single-strand RNA virus, the positive genome of the virus can be read by the ribosome and translated into the viral proteins, like the capsid proteins of the virus coat. And to make the genome, the positive strand RNA virus will make a negative strand, which is the antisense to the positive strand, and then use the negative strand to make more positive strand or more viral genome. And then the viral genome, the positive strand RNA, will get together with the viral protein to make the new RNA virion. Any questions about any of that? All right, so if the viral genome is a negative strand RNA genome, this genome cannot be read by the ribosome to be made into protein. What happens is the negative strand RNA genome will make a positive strand RNA, and that strand can be used to be translated to make the viral protein. And then furthermore, the positive strand RNA can be used to replicate the viral genome. So the positive strand will be used to make the newly replicated negative strand RNA genome. And then the negative strand RNA genome can get together with the protein coat to make the new virions. Now, if the genome is a double-stranded RNA virus, it's very simple. You make the genome single-stranded, and you use one strand to make a copy of the other strand. Any questions about viral replication? All right, if there's no questions, I'm going to move on. And I've actually already talked about this, so I'm going to skip it. And we're going to look at viral multiplication in a little more detail. We're going to look at viral multiplication of a virus that is positive sense single-stranded RNA. So the virus will bind to the cell membrane of an animal cell and be brought into the cell. The virus will uncoat, and the genome is positive single-stranded RNA, and it can be read directly, making the protein coat for the new virions. The positive strand RNA genome will be used to make the negative strand RNA, and then the negative strand RNA will be used to make more of the positive single-strand RNA genome. The positive strand RNA genome can then get together with the viral protein coat and then made into a new virion. And then these new virions will be released from the cell, either by lysis of the cell or by the budding life cycle. Any questions about multiplication of the positive strand, single-strand RNA viruses? Okay, if there's no questions, let's talk about multiplication of the negative single-strand RNA virus. Once again, the RNA virus binds to the cell membrane and then is brought into the cell. The virus is uncoated. The virus is negative strand, so it cannot be read by the ribosomes. but the negative strand, single-strand RNA genome can be copied into positive strand RNA of the virus. And then that positive strand can be used to make more of the viral genome, meaning the positive strand can be used to replicate more of the negative strand of the virus. The positive strand can also be read by the ribosome to be made into the capsid proteins, and then the capsid proteins can get together with the negative strand antisense, or the single-stranded viral RNA, to make the new negative strand viruses. And then the negative strand virus, single-strand RNA virus, can be released from the cell either by lysis of the cell or by the budding life cycle. Which life cycle it follows depends on the species of virus. All right, any questions about viral multiplication? If not, let's move on to the retroviruses. This is a family of viruses that is an RNA virus, but it does not replicate in the cytoplasm. It replicates in the nucleus. So it is an exception to the rule of thumb. And when you learn about retroviruses, you'll figure out why they replicate in the nucleus. The retroviruses include the lentiviruses, which includes HIV-1, or human immunovirus deficiency 1, or HIV-2, both of which cause AIDS, and it also includes feline leukemia virus, which gives cats leukemia, giving them a disease very similar to AIDS, and that is the cat will waste away, and when they get very sick and weak from the leukemia, they'll also get lots of infections because their immune system is shot. And it is generally a cat that gets feline leukemia virus will die, which used to be the case with AIDS. And as I mentioned, they now have medication to keep the AIDS patients alive for a long period of time. They might not live a normal lifespan, but it is, I don't know, somewhat close to a normal lifespan, so they can live with AIDS with the medication for a long period of time. Retroviruses are unique in that they contain a gene for the enzyme reverse transcriptase. Reverse transcriptase is an RNA-dependent DNA polymerase. And that should be clear to everyone what that is, right? Nobody's commenting. An RNA-dependent DNA polymerase is something, or an enzyme, that makes DNA from an RNA template. So this is one time where information flows from RNA to DNA. And retroviruses can make DNA from RNA. And that's where we get the name retrovirus. From RE, a reverse, and TR from transcriptase. So reverse transcriptase gave retroviruses their name. Any question about any of that? Oops, went a little too far. Let me blow this up. If we look at the virus here, you can see there is reverse transcriptase inside retrovirus, meaning an enzyme of reverse transcriptase. And in the genome of the virus, there is a gene for reverse transcriptase. You will also note that retroviruses are an envelope virus. They have spikes, they have a protein coat, and they have 2 strands of the positive single-strand RNA, and you don't really need to know that because I'm not going to test you on that, but they are a positive strand, single-strand RNA virus. Attachment, penetration, and coating of retroviruses follows the same pattern of other animal viruses, but their biosynthesis is different. Let me see if I can open Paint and show you. Come on. Oh, that's the wrong one. I've got to edit it. Make this bigger. Move that away. So we have a strand of RNA. Let's see if this is big enough. And RT let's see, here we go. Can reverse transcriptase, and this is RNA here. The RNA genome can make a strand of oops, wrong one. Can make a strand of single-strand DNA. And you don't really need to know this, but the reverse transcriptase will make the single-strand DNA What do you think it'll make it? It'll make it double-strand DNA. That's not drawn too well, but you get the picture. Actually, I bent it in the right place. That was totally by accident. I'm not that good of an artist. DS stands for double-strand. The double-strand DNA, and this happens in the nucleus, the RNA moves into the nucleus of the cell, and the double-strand DNA will then integrate. We'll go ahead and make an arrow again. Will then integrate into the host cell DNA. Yeah. From this stage here, the virus can replicate. Oh, let's go with that one. Not a very good arrow. Not too bad. And it can replicate RNA from the DNA, the viral DNA. And maybe I should mention that in there. host cell DNA with viral DNA, meaning they're integrated together, and the viral DNA becomes a part of the chromosome of one of our cells. And the virus can then replicate by taking the viral DNA and making viral messenger RNA. Let's move that over a little bit. Which will be a positive strand. I'm just going to go V for viral RNA. And then can use this, because it's positive strand, it can use this positive strand RNA to make viral capsids. And so the virus gets together. Sorry. And we have newly replicated, well, I'm skipping a step. We replicate the protein code, and it gets together with the replicated plus strand or single strand RNA virus to make the new virions. Over. Oh. Can't move it. Oh well, we're just going to leave it. And then this virus will bud out of the cell. So retroviruses follow the budding life cycle. And that is how retroviruses obtain their envelope. Any question about retroviruses and their life cycle? So here you can see retroviruses, the positive single-strand RNA will use reverse transcriptase to make in two steps. First they make single-stranded DNA, and then it makes double-stranded DNA. The DNA can integrate, that's not shown here, into the host cell's DNA, and that permanently becomes a part of the DNA of this cell. And then this DNA can be transcribed to make positive strand RNA virus, which is the newly replicated genome of this virus. And this positive strand RNA can be read by the ribosomes to be translated in capsid proteins. The capsid proteins can get together with the viral genome to make the new progeny virions, which then will bud out of the cell. So retroviruses bud out of a cell. They follow the budding life cycle. Let's look at that. Oops, too far, I think. Let's look at the budding life cycle. No, that's not it. There it is. In a little more detail, the retrovirus will bind to the cell membrane, and with the retroviruses, it will be these spikes, which you can sort of see. Let me blow this up. The spikes here will bind to the cell membrane, and the virus will be brought into the cell. The viral RNA will make viral DNA, which will move into the nucleus and then integrate into the host cell's DNA. Remember, when we have an animal virus that integrates into the host cell's DNA, we call it a provirus. The provirus can replicate by making positive strand RNA, which is the viral genome. And this viral genome can be read by the ribosomes to make viral proteins. The viral protein can get together with the viral genome, and then the virus can push on a membrane of the cell and pull off part of the membrane to obtain the viral envelope. And then the virus is capable of, the newly replicated virus is capable of infecting another cell. Any questions about retrovirus multiplication? All right, if there's no questions, that's it for retroviruses. Whoops, what did I do? I think I stopped the share. Trying. Oh, end the show. There we go. So let's move on to our last family of RNA viruses that I want to talk about, and that is the coronaviruses. The family name is Coronaviridae. And when we talk about the coronavirus infection, you should realize that coronavirus is the name of the family. It is not the name of the species of virus that is currently causing a pandemic in the world. Any question about any of that? The coronaviruses have been known to humans for a long period of time. The coronaviruses actually are the second most common cause of the human colds. So human colds are primarily, or the number one cause, is the family of Picornaviridae, but the number two cause are the Coronaviridae. So coronaviruses have been known to humans for a long time, and mostly, although not entirely, but mostly they cause human colds. Any question about any of that? They're the second most common cause of human colds. They are a single positive strand RNA virus. They are also enveloped. And if you notice, let me blow this up. Come on. It's not going to let me. Now it will, because I'm ready to go here. You'll notice that I've got this picture on my glass, and I picked it many years ago just because I thought this picture of a virus was really cool looking. And of course, this is the Coronaviridae. This is what COVID-19 looks like. And it just happens to be the picture I chose for our class. And I did that many years ago, long before COVID-19 ever existed, probably. We think it's a recent virus, by the way. Coronaviruses do cause serious disease in man, and we already mentioned that COVID-19 is one of them. SARS, which stands for Severe Acute Respiratory Syndrome, is a disease that initially showed up in 2003, and I think it came back later. But fortunately, I think it came back both times in China. Both times the Chinese government got control of it, prevented the spread of it, and kept it from becoming... Why is there a cross through that S? Oh, there it's better. Kept it from spreading, so it didn't spread throughout the world, and that was a good thing because the mortality rate of SARS is around 10%. So it's much higher, well, not much higher, but it is higher maybe 10 times higher than COVID-19, the mortality rate. And then MERS, Middle East Respiratory Syndrome, is another serious disease of man that is caused by coronaviruses. MERS has the highest mortality rate. Humans that get MERS, about 36% of them die. The good news is that MERS does not spread from human to human. All the people who have gotten MERS have been in close contact with camels. So apparently MERS is a virus that camels have, and occasionally it crosses over from camels to humans. And when humans get it, it's very serious. Like I said, about 36% of the people die. With COVID-19, the mortality rate is estimated to be between 0.2% and 3%. But it does spread more easily than the flu. So COVID-19 spreads very easily. In general, when one person comes down with COVID-19, if they're around unvaccinated people or unimmunized people, they are likely to infect 1 to 2.5 other people. And that is why COVID-19 spread so rapidly, as well as the fact that there are a large number of people who get COVID-19. The Center for Disease Control estimates that at least 1/3 the people who have COVID-19 are asymptomatic. And among young people, like the majority of you and the majority of teenagers and children, about 50% of them will get COVID-19 and they will be asymptomatic. And when they are asymptomatic, they don't know they have COVID-19 and they can spread it to other people. Any question about that? Okay. COVID-19, by the way, got its name because it is a coronavirus, and that's what COV stands for. And then it was identified in 2019, and that's why it's ID-19. So that's where COVID-19 got its name. It does have another name, and it's a scientific name, or a family name, whatever you want to call it. It is called Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. The trouble is, this name is so similar to SARS that even among the scientific community, most of them call it COVID-19 because they don't want to get SARS confused with SARS-CoV-virus. I'm saying that coronavirus 2. COVID-19 is closely related to bat coronaviruses and likely originated from bats. They believe that COVID-19 is actually a recombination of 2 other bat coronaviruses and 1 pangolin virus. So it's a recombination of 3 different coronaviruses. The pangolin only accounts for a small percentage of the genome. I think it might be about 10%, but it codes for the spikes of coronavirus. And then the rest of the coronavirus is encoded by bat coronaviruses. They think that COVID-19 evolved in bats, meaning that's where it recombined, and then it jumped to an intermediate, perhaps the pangolin. If you don't know what a pangolin is, it is a spiny anteater. And then they think it spread to humans. They're not sure, but they think that this happened in China, and the Chinese like to eat a lot of exotic animals, including pangolins. And in the Chinese market, what is that, Wuhan? They had pangolins in the Chinese market. And they think that one pangolin had COVID-19, and it jumped from the pangolin to a human, and then COVID-19 started spreading in Wuhan, China. Now, this is not known for sure, but this is what is most likely how COVID-19 came about. A recombination of 3 coronaviruses that probably happened in bats, and then jumped to the pangolin. The Chinese like to eat exotic animals, and in their animal markets, like in their animal markets in Wuhan, they had a whole bunch of exotic animals for people to buy and then eat, and they think there were pangolins there. They think a pangolin had COVID-19, and COVID-19 jumped from the pangolin to the humans, and then COVID-19 spread among other humans. Now, let me mention that they have not found COVID-19 in the Chinese animal market in COVID-19, but it may have only been in one pangolin, which was probably bought and eaten in 2019. So that might be why they haven't found it in the Chinese market. Anyways, the Chinese have now learned that 3 times they have gotten a serious disease in their animal markets, COVID-19 and SARS, and I don't remember if the 3rd time was also SARS or what, but they've gotten 3 times a serious disease coming out of their animal markets, and they've decided to make it illegal to sell exotic animals because it appears that the Chinese animal markets were a good place for viruses to jump from an animal to a human, and then humans get a very serious disease. And hopefully that will reduce the chance of a serious disease like COVID-19 happening in the future. Any question about any of that? All right, COVID-19 is transmitted primarily by droplets from cough and sneezes within a range of about 6 feet. It may spread further, and it may spread from indirect contact, like when somebody coughs or sneezes and the virus gets on a surface, and then people can pick it up with their hands and then touch their hands to a mucous membrane in their face. But it seems like COVID-19 is primarily spread via droplets, mainly from coughs and sneezes, although it could be from breathing or singing, of a distance of about 6 feet. And it is surprisingly rarely picked up by the hands and carried to the face, because the virus can stay alive for about 3 days on plastic or metal surfaces, on 1 day on paper or cardboard. And so when I get my mail, I always let it sit for at least a day before I open it to make sure if there was COVID-19 on it, that it'll die before I open it. Any question about any of that? It's surprising that COVID-19 isn't picked up by people's hands, because with the flu, that is primarily the way people get infected with the flu. They pick it up on their hands, like touching a doorknob or a water faucet or a light switch, and then they self-inoculate themselves by touching a mucous membrane. That is the majority of the way that people get infected with the flu. But COVID-19 does not seem to primarily spread that way. It can spread that way, but primarily it's breathed in. So that doesn't mean you shouldn't worry about it. You should take precautions when you're touching surfaces that may have COVID-19 on them. Some symptoms of COVID-19 include flu-like symptoms. Though runny nose and sneezing are less common, and shortness of breath and deaths are much more common with COVID-19 than with the flu. The mortality rate of COVID-19 increases with age, with the ill health of the patient. It's more common in men than in women, meaning men are more likely to die of COVID-19 than women. And then for some reason, blacks and Hispanics have a higher mortality rate when they get infected with COVID-19. Any question about that? I don't know why blacks and Hispanics have a higher mortality rate, but in this country, it is very obvious that blacks and Hispanics have a higher death rate than whites and Asians. The incubation period, a person gets infected, and then that virus will incubate in them for 2 to 14 days post-exposure. Some patients remain asymptomatic, and that's a high number. It's estimated that at least a third of patients are asymptomatic. Among young people, it's 50% of the patients are asymptomatic. As I stated, the death rate is estimated to be between 0.2% and 3%. The actual number as of May 29, 2021 is about 1.8% of people who get COVID-19 in the United States will die of it. And this number differs for different countries. Mexico has the highest death rate. I think their death rate is close to 9%. And then some European countries have, and also some Middle Eastern countries, have a much lower death rate. And we're not really sure why some countries have a lower death rate than other countries. It's pretty obvious that the healthcare in America is better than the healthcare in Mexico, and so that's why we have a lower death rate than in Mexico. But there are other factors coming into play. Obviously, there's more Hispanics in Mexico than there are in the United States, so that would be another reason why the death rate would be higher in Mexico than in the United States. For prevention, use correct hand washing. When you wash your hands, you want to do it everywhere on the hands and wash for 20 seconds before you rinse the soap off. Another thing you can use is sanitizer. It's very good at washing your hands. The coronavirus is very susceptible to the alcohol in sanitizer. Keep your distance from people, especially when they're coughing. Don't touch one's face. Wear a face mask. And then keep your numbers in low, meaning keep your people that you're around in low numbers to prevent the spread of coronavirus. And of course, the best way to prevent the spread of coronavirus is get vaccinated. That wasn't possible until recently, or fairly recently, but it is possible now. If you're sick, you always should wear a mask so you don't give the coronavirus to someone else. It's best to stay home when you're sick and cough into your elbow or into tissue, and then wash your elbow and throw the tissue away. All right, I think I will stop here and we will continue with this next time. Are there any questions about coronavirus? Oh, I already got that marked to here, so just save it. Any questions about anything we've discussed today? If there's no questions, I will see you at 6:30. If you have a question from the lab, if you do, make sure you log in before 6:45. I will see you.